Hello everyone. Topic for today is Histidine Operon. So before getting into the topic, let's know what's histidine. So histidine is an amino acid, and as we know, amino acids are used to make proteins and enzymes in the body. It is sometimes referred to as semi-essential amino acid because it is non-essential in adults but essential in the diet of infants and those with a kidney disorder called uremia. So, histidine is also called L-histidine and alpha amino B 4-imidazole propanoic acid. So, if we talk about the function of amino acids, the function of histidine. So, histidines are used by the body to make specific hormones and metabolites that impact kidney function, transmission of nerves, stomach secretion, and the immune system. And histidine also has an impact on the repair and growth of tissues, making blood cells and helping to protect nerve cells. It's also used to make histamine in the body. So if we talk about one more function, which is let's say the primary function. So the primary function of histidine in the body is to regulate and help metabolize and break down the use of uh, breakdown and use for energy. These it also help in metabolizing trace elements such as copper, iron, zinc, manganese, molybdenum, and more. So now we'll get into our topic, which is histidine operon. Before knowing what's histidine operon, let's know what's an operon generally. So operon is a genetic regulatory system found in the bacteria and their viruses in which genes coding for functionally related proteins are clustered along the DNA. So this feature allows protein synthesis to be controlled coordinately in response to the need of the cells. So a typical operon consists of a group of structural genes that codes for enzymes involved in metabolic pathway such as the biosynthesis of amino acids. So these genes are located on a stretch of DNA and are under the control of one promoter. Also, a single unit of messenger RNA is transcribed from the operon and is subsequently translated into separate proteins. So the promoters are controlled by various regulatory elements that respond to environmental cues. One common method of regulation is carried out by regulator protein that binds to the operator region, which is another short segment of DNA found between the promoter and the structural genes. So this was a small short note about uh, operon. Now moving, so this is the structure of simple operon. For the sake of this video, I've taken histidine operon. So we can see the sequence, the repetition of histidine. So now let's talk about histidine operon. So this histidine operon is mainly seen in the bacterium Salmonella typhimurium, and it controls the rate of histidine biosynthesis, and the biosynthesis of histidine in Salmonella typhimurium, and also it is synthesized in E. coli. It has been an important for the system. It has been important system for the study of relationships between the flow of intermediates through biosynthetic pathway and the control of the genes encoding the enzymes that catalyze the steps in pathway. So the earlier studies of histidine biosynthesis contributed to understanding mechanism basic to the regulation of biosynthetic pathway, such as feedback inhibition, energy charge, and setting of basal biosynthetic enzyme levels. The histidine biosynthetic pathway itself contains several interesting and unusual intermediates and enzymatic steps and forms a critical link between amino acids, purine, and thymine biosynthesis. So this this histidine operon in Salmonella typhimurium is believed to cause toxicity to food materials. So food companies, before approving their food products, they undergo a small test which is called as AMES test or AIMS test, which helps in getting the knowledge if the product is pure or not. So the AMES test, the objective of this test is to determine the mutagenic activities of chemicals by observing whether they can cause mutation in sample bacteria or not. So these bacteria are usually kept in petri dishes, and they are some bacteria are provided with the nutrients, let's say histidine, and some are not. And also they are provided with chemicals to see such bacteria can use the chemicals and synthesize histidine or not. So the principle of this test is that this test uses several strains of bacteria, uh, let's say Salmonella typhimurium strains that carry a particular mutation. So these Salmonella typhimurium bacteria that are taken for the test are mutated strains. They are not the natural ones. So the mutation that's done is a point mutation. Point mutation meaning the a single mutation is done at a target site. Let's say in Salmonella we are point mutating the histidine protein, the histidine or the tryptophan. So these mutations in the histidine will inhibit the organism from growing until unless histidine is supplied. So we look at the process. So a sample mutagenic potential is assessed by exposing amino acid requiring organisms to varying concentrations of chemical and selecting for the reversion event. Media lacking the specific amino acids are used for this selection, which allow only those cells that have undergone the reversion to histidine. The reversion to histidine to survive and grow. So if the test sample causes this reversion, it's a mutagen. So by this test, as I said, we can determine whether a chemical is mutagenic or not. So this was a small uh, illustration about histidine operon and how AMS test helps in determining the mutagenicity. Thank you. Uh, Make sure you subscribe to the channel for helping us to create such videos. Thank you.